Hello everyone and welcome back to Simply Biology. In today's short video we're going to be focusing on protein synthesis. So what we're going to be doing is we're going to be looking at the mechanism for protein synthesis, which involves both transcription and translation. Now without further ado, let's get started. So first step, DNA helicase, which is an enzyme that's also involved in DNA replication, if you remember rightly, unwinds the DNA strand by breaking the hydrogen bonds between the complementary base pairs. Now remember, between A and T there's 2 hydrogen bonds, and between C and G there's 3 hydrogen bonds. Now this enzyme unwinds the DNA by breaking those bonds. Then RNA polymerase binds to the template strand or the non-coding strand. So don't get this mixed up with DNA replication, whereby it has DNA polymerase. In protein synthesis, which is this process here, RNA polymerase is the enzyme. And this binds to the template strand, which is also known as the non-coding strand. The RNA nucleotides in the nucleoplasm are activated by the addition of 2 phosphate groups from ATP. So again, very similar to the process of DNA synthesis, but it's RNA nucleotides instead of DNA nucleotides. So these activated RNA nucleotides diffuse through the nucleoplasm in a line next to the complementary base pairs on the template strand. So what happens is the RNA polymerase then joins these adjacent nucleotides together by forming covalent bonds known as phosphodiester bonds. What happens is then this process continues until all the RNA polymerase reaches a stop codon, which is a specific codon that halts DNA replication or halts protein synthesis, and it codes for stop. It doesn't code for an amino acid. What this does is it causes the mRNA strand, which is also known as messenger RNA, to fall off and fall out of the template strand. The process of the production of mRNA is now complete. Right. Once mRNA is complete, translation can occur. So the mRNA molecule, abbreviated for messenger RNA, can move out of the cytoplasm or move out into the cytoplasm, and where it joins to a ribosome. This ribosome can either be free in the cytoplasm or can either be attached to the rough endoplasmic reticulum. So the ribosome reads the mRNA strand, and this is done by the smaller unit of the ribosome, whereas the larger unit of the ribosome is used to join the amino acids together. But what happens before that is it's read by the mRNA strand, the small unit, remember, and tRNA molecules with complementary anticodons bring amino acids. So there's something called a tRNA amino acid complex, and that is simply just a tRNA molecule, which is an RNA, that brings the amino acid to the ribosome. And what happens is that the ribosome reads the amino acid, the codon sequence, the mRNA sequence, and it selects a tRNA molecule with a complementary anticodon to that codon sequence. What happens is that tRNA molecule then binds to the mRNA sequence, and the next tRNA molecule binds. So what happens is the ribosome then joins those 2 amino acids together, and the tRNA molecule is free to release back into the cytoplasm to collect another amino acid. So this process continues to form a polypeptide chain. So when the ribosome reaches the stop codon, it falls off the mRNA and the synthesis of the protein is complete. So then what happens is this protein can fold to form a secondary structure, tertiary structure, and the protein can have multiple functions such as antibodies, enzymes, hormones, or any other process in the body. and that is the process of protein synthesis. I hope you enjoyed this video, and thank you for watching. Goodbye.